Okay, good morning, everyone. So I'm Purya Dasme. I'm a postdoc at the University of Zurich here in Erchel campus. And I'm also affiliated with Harvard University. And in this part of the course Bio 390, my aim is actually to introduce some of the useful concepts in biological networks. So the way that we achieve it is that we have 2 sessions of 45 minutes. In the first session, I'll go through examples of biological networks, and then in the second one, we'll review some of the properties of these networks. And I'll have a chance at the end of the lecture to just talk about the sort of questions that you might get in the exam, which are not tricky questions, so the aim is not really to surprise you, but the aim is actually to make sure that we understand some basic concepts. Okay, session 1. So right away, as I told you, this is an interactive lecture. I already have a definition of a network, but I'd like one of you to volunteer and tell me what is a network. What do you think a network is? I'd simply like you to start, discussing and talking. Who would volunteer? We won't actually move forward until one of you speaks, so you can stay here 45 minutes, I guess. What is a network? Go ahead.
So different entities are like communicating with each other or are part of each other. So
Perfect.
A does something for B and B something for A.
Perfect. That's actually a great answer. We have different entities, and these entities either communicate or interact with each other. And the key word here is some sort of a relationship or communication. As we have here, there are different nodes. So what happens is that we need actually to define certain interactions between these nodes to call this set a network. So we cannot basically call, networks that are not communicating with each other. We might actually have certain types of networks with very weak communication, but in principle, you need actually a bit of interaction between these nodes and entities. What is the interaction, of course, is the big question, and it really relates to your field of interest. So if you're an ecologist, interaction could be relations between species. If you are a protein biochemist, interaction could be electrostatic interactions, hydrogen bonds, or whatever. Biological networks are networks where these entities and nodes are biological entities and biological systems, right? Anything that would have biological meaning would actually be part of this biological network. So we can think of these nodes to be DNA, protein, cell, individual organisms, communities, and so on and so forth. So perhaps the simplest network that we can think about is simply biomolecules. So we have DNA and protein. And the way that molecules or amino acids are connected in these macromolecular structures would define the network. However, the nice thing about network is that you will learn, I guess, through this lecture and maybe from the past experiences, that if you change your point of view, you might actually get a different network. So networks are sort of multifaceted. So here in this case, if I'm interested simply in the chemistry, if I'm interested in the interactions between the connection between different amino acids, for example, how this amino acid here is connected to the other one, I'm actually thinking about a chemical network. But we can also define a different network, which is sort of the 3-dimensional network. And this is the contact map of the protein. So you can actually think of the chemical structure, a bunch or a set of chemical bonds that would connect different atoms. But then you can also define a contact map. Who knows what a contact map is? Go on.
The single amino acids mapped out and how they touch each other. So the structural information is given there.
Yeah, so good point. This is actually a good definition, but if we just come to the plot here, so there are different dots here, and then the majority is sparse, so you don't have many dots. But what's the meaning of each dot? Yeah, go yourself.
Where they touch or how they maybe interact with electrostatic or hydrostatic
Good, good. So basically, the meaning of each dot is proximity. So if two amino acids are close to each other, we put a dot on this map. And as you said, many of such dots would implicate interactions. So you have dots because some things actually like to come together. Otherwise, they would have been unfolded. So there's actually a force to keep proteins folded. A thermodynamic, basically, ground state would prefer such folding. So the reason that I actually put this slide is to tell you that if you have a bunch of entities, you can always think about different ways to characterize this network. So one is simply the connection between them, and then the other one is closeness or proximity. So the first interesting or biologically interesting network that we come across quite often in computational biology is, or experimental biology, is basically protein-protein interaction network. So here, each node would represent a protein, and edges in this network would correspond to interactions between these nodes or proteins. So we might have complexes here, meaning that there are many proteins that need to interact with each other. We have hubs. Some proteins would interact with more partners. We come across the definition of hub quite often through the lecture. But just this is an example of a basically protein-protein interaction network within a complex. So this is Pol-2 complex, RNA polymerase II complex. And we see these are different subunits, and they can interact with each other. So have you worked with the string database before? Good. So what I would like you to do now is open the string database on your laptops or phones and try to look at two networks, the network of interactions with myoglobin and its partners, and the network of interactions with 4 DNA-directed RNA polymerase and the partners. So you can actually open the browser here and then put these two words and then see what you get. It's very simple. So this is actually built to be user-friendly. So you just need to open and put it. Is anyone having problem addressing or finding it? I guess it was very easy to get it. Right. Find. I guess you did this already, right? Okay, so very simple thing to do. Now the question that I have is, what do we learn from this? Anyone wants to say something? What are the differences between, let's say, this network and the other one? While you're thinking, I'll explain something else. You can also do something quite interesting on the string database. You can go to setting. You can actually indicate that, so the first shell is no more than 10 interactions. So you only need 10 interacting partners. And you can also define the second shell. Okay, so the network gets a bit bigger here. We can do the same thing for myoglobin. Okay, so what is the difference between these two? Just say something. Yeah, go on.
I guess in the RNA polymerics contact map or the interaction network map, we can see so many very complex. They are creating complex.
Absolutely. So this is actually, so the most obvious information that you can get is basically number of interactions. So some proteins interact more with other partners. But the interesting thing is that if you look at this website, so in the setting, you have actually the definition of these interactions. So you see to start from here. Let's see. Okay. So basically, we have the definition of interactions. Some of them might be coming from text mining, experiments, databases, co-occurrences, co-expression, and so on. So it means that we can define a network based on different biological meaning and biological experiments. So we can do something fun here. So let's say I'm just interested in experiments. So somebody in the lab observed by fancy and interesting methods that two proteins would physically interact with each other. So actually tick off all these guys and keep the experiment. Can update this map. Now we have a lonely myoglobin here. So myoglobin hasn't been found so much to interact with other proteins. But if we go to the RNA polymerase II and do the same thing, still there are so many partners that are there. And these physically interact with each other. So how would they achieve it? This is a question, of course, for biochemists. They can be different, a wide range of interactions, electrostatics, hydrogen bonding, and so many other things. Okay, so let's go back to, thanks for checking this with me. Yeah, so these are the two examples that we talked about. So it brings me to the next question, which I believe is a fundamental question, and the question is this. How come we have different tissues and cell types, but the reality is that we have only one DNA molecule? Do you find this question fundamental and interesting? So imagine we have only one molecule, so this is the DNA molecule, but somehow this DNA molecule in some cells would give rise to different functions and cell types and morphologies. Who has any clue on that? Go on. Good. I like the keyword gene expression, but we need to add something to it. Perfect. So this is actually, in combination, this is the perfect answer. So the reason is that we have... Okay, so the reason is that we have a network of interactions, which we call gene regulatory networks. So the way it works is that we have different genes. So here I'm showing gene 1, gene 2, gene 3. Gene 1 would actually transcribe mRNA, would be translated to protein 1. So this might be a transcription factor, binding the promoter of gene 2, changing the expression of RNA2, which is basic mRNA number 2, leading to protein 2, and then basically finishing this cycle. And here what we see is that this actually defines a gene regulatory network. So if in different cell types we have different gene regulatory networks, That would be the reason that we get different types and functions and morphologies, which is a very interesting question. Okay, so the second fundamental question is, well, cells have this extraordinary ability to respond to stress. So if you don't provide food for yeast, for example, it would actually stay dormant for many years, actually. How cells achieve this coordinated response? So how would it be possible that the cells would feel the environment and basically fine-tune their survival to optimize and maximize their growth, basically? Yeah, that's a good point. Two pathways are involved here. So one is, on a more fundamental level, is a gene co-expression network, and that's the situation when we have different genes that would co-express together. So you can imagine that if we look at this network of protein-protein or gene-gene interactions, we might have sub-domains or modules within this big network, where this module is simply there to feel the environmental stresses. Whenever there is, for example, a stimuli in the environment, so that would lead to the expression of some transcription factors, and that would activate a module that is basically responsible for survival under stress. And so the way that we can actually get these networks is often through experiments. And I'd like to make a comment here because if many of you would end up becoming computational biologists or bioinformaticians, please do consider reading and knowing a lot about experiments. So don't just believe that the aim of the experiment is to provide numbers, because the more we know about the experiments, the better we can design the analytical procedures. Anyway, so we have RNA-seq and microarray experiments. So the aim of these experiments is to estimate the expression level of different genes. Here I'm showing from gene 1 to gene 10. And we can imagine different conditions, S1, S2, S3. So we have a vector with 3 elements here, another vector here. For each gene, basically, we have a vector. And then we can calculate the correlation between these expression levels. So, of course, the correlation between a gene and itself is always 1. But we can actually get different numbers in this matrix. What we can do, we can assume that when correlation is above certain threshold, 80%, we call it a co-expression. And this can be arbitrary, of course. But 80%, 90% could be a good estimate. And that actually would lead to generation of a sort of binary matrix here. Sometimes we call it network adjacency matrix. So adjacency matrix. So what it means is that we can actually go in this matrix, find the elements that are 1. So it means that gene 1 and G7 would co-express together. And from this binary matrix, we can build a network. And this is very important. So whatever network that you observe, there's a 1-to-1 correspondence with the binary matrix. That's actually very important and useful because later on, if you have a network or whatever this network is, you can construct the corresponding matrix, and then you can use linear algebra on that. You can calculate so many interesting properties. Another set of networks, or another biological network that we are often interested in, is metabolic network. So we know that, so basically metabolic networks would contain chemical reactions that can either generate energy or build essential molecular blocks, for example, DNA and protein and amino acids. So building blocks, basically amino acids or nucleotides. And these reactions are basically catalyzed by enzymes that are themselves coded by genes. So when you look at the metabolic pathway or map within a cell, it's often very complex, and it looks like a sort of blueprint or subway map, which we'll come to that later. But basically, we have different nodes, different arrows, and then we can think of different directions. What are these entities? Let's practice a bit together. So have you worked with the KEGG database? Good. So basically, this is Kyoto Encyclopedia of Genes and Genomes. And what you can do is that just search KEGG pathway database and try to find the relative pathway for folate biosynthesis. So you find this guy, then put folate biosynthesis, and then see what you get. So could anyone get this picture? Good. And the rest are looking at it. Great. So in these, so this is the corresponding metabolic pathway or map for folate biosynthesis pathway. So you have nodes here that would correspond to metabolites, like folate. And you have arrows here that are reactions, and then on top of the arrows, there are boxes here that would show the enzymes with their sort of EC classification. So the reason that you might actually work with this EC number or know about it, but usually we try to classify enzymes based on their chemistry. Okay, so one interesting thing is that you can compare folate pathway in different organisms. That's what we want to do now. So we want to look at folate pathway in E. coli and compare it with the same pathway in human. So do that, please. So there is a tab up there that you can choose the organism, and then just see if you can change the pathway from E. coli to human or the other way here. So when you do that, you should ideally get this picture. So you change the organism, and then you can get two different maps. So I need actually one of you now to tell me what are the differences between them. So preferably anyone who hasn't spoken yet and thinks that his or her answer is wrong. We learn by mistakes, we don't learn by just perfectness. Just to make life easier, you can just look at these things, right? These two maps. There's no need to look at the screen. So what's the first difference between them?
Some of the labels are green and some are blue.
Perfect. This is the perfect answer, actually. Did you think about this answer and thought that maybe it's wrong?
I thought it was too simple.
What I mean, the purpose is to make things simpler. Okay, so exactly as your friend said, the difference is that there are different boxes. So the topology is the same, right? These are the same pathways. But the difference between E. coli and human is that some of these boxes are green here, so there are basically more green boxes in E. coli compared to human. And let's go to the folate. Folate is here, right? And particularly what we see is there are many green boxes around folate, and it seems that, well, the flow, or basically the set of green boxes, never ends, right? So you can actually have all green boxes, green boxes until you get here. But if you look at the human, so the metabolic pathway for human, you see that, yeah, it seems kind of sparse, right? So we have some green boxes here, but we don't have many green boxes up there. So what can we learn from this? Green means presence of an enzyme. Well, if you don't have the final answer, it's okay. Just give the intermediate answer. Go on, please. Some comments.
I would assume that the pathway is faster in E. coli because the enzymes catalyze the reaction.
Interesting. So the fact that you say it happens faster is important. But we are not comparing rates here. So what else the lack of these green boxes mean?
I mean, in humans there could be other pathways which do the same job as in E. coli.
Interesting. Okay. You go, please.
Maybe because there aren't too many boxes which are green around folate. Well, we can't make folate.
Perfect. Perfect answer. Great. So the reason that we don't have green boxes around folate is that we cannot actually synthesize folate de novo, so we need to take it from nutrition. That's the reason, usually parents tell us to eat leafy vegetables and so on and so forth. But E. coli has this ability to synthesize folate de novo, meaning that from the scratch, starting from some other metabolites, and then basically synthesizing and getting folate. So just by looking and comparing these two pathways, we can learn a lot about the differences between species. So that's one source of information. So E. coli basically belongs to prokaryotic organisms. Many of such organisms are pathogens. So we can actually learn something more interesting. Knowing that folate is actually a very important metabolite for growth, so we need folate to have cell growth, it means that if we cut this pathway at some point by using some inhibitors or whatever, E. coli actually loses the ability to synthesize folate. Humans are fine because they can actually get folate from nutrition. And that is one recipe to design antibiotics. So there is actually a great class of antibiotics that are called sulfonamides. And what they do is that they right attack this enzyme. So we design certain enzyme inhibitor, DHFR inhibitors here, which is actually the enzyme here, that would inhibit this step and would actually lower the production of folate in the cell. So just by looking at these networks, we can sort of even come up with the strategies that would be interesting for medicine. Another useful concept is that when we look at many pathways, but especially metabolic pathways, people often draw similarities between such pathways and subway maps. So here I have a metabolic pathway of a certain pathway, a metabolic map of a certain pathway in E. coli, and then here this is New York subway map. So the question now is, what are the similarities between these two? Again, preferably somebody who hasn't spoken and thinks the answer might be wrong. Good. Great. Great. This is a great point. So you can. So what you mean is that you can get from one point to another. So you won't be lost somewhere. So that's actually. So there is. So this is a concept of flux. So you can actually get from one part to another. That's the first similarity. The second similarity is the presence of certain hubs, right? So here, for example, in this case, maybe this is around Fifth Avenue, for example. Or so what you see is that there are hubs. So these hubs are important stops in the city because many people want to visit, or in this case, these are important metabolites because it's needed to be catalyzed and to process the cascade of events in the cell. But the third difference that I actually like to mention is something that we call, and you know already, is the concept of a detour. So basically, you can get from one part of this network to another part by different routes, right? So you can actually take 2 or 3 different routes to get from one node to another. The same would happen here. So imagine if you want to get to Milchbuch from Hauptbahnhof. So what happens is that you don't only need to take one route. You can go to, Altstetten, you can go to Stettbach, and then come to Milchbuch. What does it mean, the fact that we have different routes or detours in a network? Is it a good thing or a bad thing? In the most simplistic way. Well, that's correct. But what I actually like to think about, so I like actually the word adaptation, and adaptation would require such a thing. But let me introduce a word, because maybe the word is not that familiar now, and then we talk about it. How about robustness? Which system is more robust? When we say robustness, it actually means the stability in the face of change. So if something happens to tram number 7, well, you have other buses to come here, basically. So the same would hold and would be true for metabolic networks. So usually the way that metabolic networks are built and evolved, which is the better word for it, is that it actually encourages and promotes the maintenance of robustness in the system. So your system is robust. So if something happens with one metabolite, there's always one other way to probably synthesize that. And organisms can use this strategy for adaptation, as your friend said. So I guess we already talked about different communications in the cell. In addition to these networks, we have also something called signaling networks. And this is when we have stimuli in the environment, and the stimuli can act on cell surface receptors. And these receptors would initiate a set of reactions and interactions that would finally govern the gene expression. And that's actually, if you really think about it, each cell has this ability to communicate with other cells. And that to me is really fascinating. I don't actually ask you now to check this database because of time, but you're just welcome to look at it at home. So you can actually look at this database called Reactome. So in Reactome, you can put a pathway of interest. So one fun thing to do, you can actually put COVID-19, for example, and then learn about pathways that are involved for the immune response to infection, and so many other things. And learn what are the nodes, and what are the connections, and what are the enzymes and proteins. And many of such databases have the abilities that you can also do analysis. So consider that for your projects and for your questions that you might have in mind. And a great part of this business in bioinformatics and computational biology is to be able to do analysis at the end. So which, after learning about them, you can actually ask many questions and then check them here. So we talked about several networks. So do you have any other network in mind? Any biological network that might be interesting to just mention? Any idea? What could be? Yeah, go on, please.
The immune system.
Yeah, great. So it is a network, but more or less is within the type of, or the definitions that we had so far. What I'm thinking is, so if you think about the sort of the order of networks that we talked about in the lecture, was we started with molecules, genes, proteins, interactions between genes, so and also cell-cell communication. So what's bigger than a cell?
Ecological networks.
Yeah, you also have the same. Good. So ecological networks, basically. So we might think, for example, there are so many of these networks. One of them is food web. In food web, you have different nodes corresponding to different species. You have red nodes here, for example, that would determine basal species, such as plants. Orange nodes intermediates, and then yellow nodes that are on top are top species or primary predators. So this is another network that would be interesting. So we have, this is the end of session 1 of the lecture. And in session 2, we talk about properties of networks, but let's have a 10 minutes break. And we'll start around 10 to 9. Okay, so let's start session 2. So we'll have, I hope the first session was informative. We'll try to have more fun in this session, hopefully. Okay, so we'll talk about properties of networks in this session. So we'll use a concept from mathematics to better understand these properties. Nothing, would at the end of the day be highly understood if you don't use tools from mathematics or calculations and analytics. So simply, we represent networks by graphs. It's very actually, you have seen different graphs. In graphs, we have nodes and edges, and these nodes could be connected with different edges. So basically, we can actually think of a graph as two sets, the set of nodes and the set of edges here. We might have different types of graphs, so unidirectional graphs, where the information goes on from one node to another, unidirectional, or bidirectional, meaning that the information might actually go come and back. So if there's an equilibrium or there is some signaling that can go back and forth between two nodes, we can use a bidirectional graph. So most of the time, here we are dealing with unidirectional graphs. We can define paths on a graph. So path is basically, a path is a set of edges that would connect two nodes. Well, simply, what you can see is that there are different paths that would connect different nodes. And that, for example, in this node, you can actually go to the other node with this path. You can also take another path. But the number of edges that would be incorporated, or the number of edges that you pass to get to the second node, is the key difference here. So if a path between two nodes is the shortest path, we call it a geodesic, basically. So geodesic is the shortest path between two nodes. These are just definitions, but we'll come to some activities later. Again, we encountered this when we were talking about co-expression networks, that a graph can be represented by a matrix, and that's what you see here. So so each node here would actually correspond to different rows and columns. So here is node 1 and 1. So you see there is no connection to itself. So if we have a self-connecting node, we basically should have an arrow like this, right? We don't have it, so there's no connection. There is a connection or a path between node 1 and 2, so we put 1, nothing between 1 and 3, 0, 0. And then here you come to the point that there is a path from node 1 to 6 and 7. So we can build a binary matrix from these graphs. The first important thing about any graph is basically the idea of degree distribution, or what is the degree of each node. So that is simply the number of edges that go into that node, or the number of neighbors that this node is connected to. So here, for example, node 1 is connected, or actually is linked to 1, 2, 3 other nodes. So the degree of node 1 is 3. The degree of node 6 is 1, 2, 3, 4, 5. Degree of node 4 is just 2. So the very basic property that we often use in describing networks is the distribution of these degrees. What's interesting, just to bring one example from biology, is that if you have a node, which in our case is a protein, and this protein is connected to more partners, would interact actually with more other nodes, usually the rate of evolution of that protein would be much lower. So here on the x-axis, we have number of protein-protein interactions. On the y-axis, I have substitutions per site. So what you see is that the number of interactions basically would determine the rate of evolution. Okay. And that's actually one interesting concept, because just by looking at the degree of each node, we can assign meaning, and this meaning here is actually the rate of evolution. Okay, so we have an activity here just to get more of this from this important concept. And I want you to spend some minutes and determine the degree distribution of your network of friends. So what you can do, you can assume that you have 5 friends, and just put yourself. So each person would be a node in this map, right? So let's say this is me. This is friend number 1, friend number 2, and so on. So consider 5 friends, and then draw whenever those people have a friendship. You just want to see how this network would look like. Is the task clear? I guess. We'll all have more than 5 friends, hopefully. So, So does anyone have a picture now for the graph of friendship? Not yet? Do we have a network? So at the very least, you should have 5 points that are completely isolated, I guess. So is anyone done with the exercise? Okay, did I need one person to come here and basically draw the network here for us? Please. You're done with it. Yeah, come. Thank you. Yeah, here or there. Just we want to see how this network, how does it look like? Interesting. So, does anyone have a different network from this? So all of you just look like this exactly, or? I mean, you should have a different network. Come on. So anyone who thinks his or her network is different, just come and put another one here for me. This is a nice network, so. We're just digesting what's happening, so please collaborate. Thank you. Good. So do you have 5 friends that are highly connected? Oh, that's perfect. So this is a subgroup, basically, a group, a perfect group. So that's actually a good point. So if you look at these 2 graphs, so what we learn is that we might have different types of graphs, types of connections, types of friendships, whatever we call, and that could be represented by a network. And so, well, if you believe it or not, your whole life might be simply a function of this network. So what I mean is that the nodes could have different connectivities, and despite the differences, we have also similarities. What are the similarities? What's similar between these two? On a face value, you just zoom out and then try to look at it. What seems similar between the two?
Number one is connected with everyone.
Yes, exactly. So we have hubs again. So this hub is occurring. So we have number 1 here in that friendship network, of your friend. And then the other one, let's call it number 1 also. So these 2 persons are apparently, but this is actually the same for all of them. So we have 5 connectivity for all of them. But then here we have the hub here. Another thing that sometimes in network, both in network theory and network biology, we consider is the presence of motifs in networks. Just have an eye or have an sort of intuition about them. So we might have a triangle, right? And this triangle might happen, in many places. And so the presence or the frequency of these motifs might have specific meanings. We have that explicitly in biology when we are interested in higher order interactions. Do we have a case? Do we have some species that would interact, with basically 3 other members, and they also interact with themselves? Do we have such a scenario or not? And these things happen quite often in biology as well. Great. So since you do computations and you use coding a lot, there's actually a very nice package that you can use for visualization and also calculations on graphs. And it's called iGraph. So here you can build so many networks with different properties and connectivities. So here you see I'm basically showing a network with 10 nodes. And then I'm specifying the degree to be 2, so we get this. Specifying the degree to be 3, we get this network, and then 4, the other network. So you can run it on different systems and with different languages, whatever your preferred programming language is. So now that we learned about degree distribution, one interesting point is that degree distribution can distinguish different networks from one another. So we have, as the null model, we have a random network. I'll come back to it as an activity later, but this is actually simply a network where nodes are randomly connected. And we can get from random networks to other types of networks. So here, for example, this is the case of scale-free networks. So the difference is basically in their degree distribution. So here is more of a Gaussian distribution. And this is more of an exponential or a power law distribution. So different distribution would entail different graphs. So an interesting question is basically whenever you get a network is to draw degree distribution, this probability, and then see how does it look like. Would it actually look like more like a random graph? Or we might have some structures like scale-free networks. Now we do another activity. I talked about random graph. So usually we use randomness as a null model, right? How can we build a random graph? Anyone has any idea how we can build a random graph? So the name implies it's random, right? So something should be random. What is random?
The degree of each component,
Connectivity, okay. So the same or good. This is true to some extent, but there are actually 2 very delicate concepts that I'd like to elaborate on. Because if you just assign connectivity randomly to a graph, it won't be a random graph. So randomness, go on please. Yeah, exactly. So both the location and the probability of forming a bond is drawn randomly. So to do that, we want to do an activity, and I need 2 friends to help me. So one friend would flip a coin, and the other one would roll a dice. Okay. So there are 6 numbers here. Let's do this. So who wants to actually be the roller, and who wants to be the flipper? Think about it until I get this done. Okay, 6 nodes. Who wants the dice? Be brave. It's not poisonous. Who would flip a coin? a simple thing. You hold it and then you do it like this. Okay. Do you want this or you have? Okay. Since I have already. Thank you. Okay, perfect. So what's your name?
David.
David. So David would tell me two numbers. What's your name? Turian? Florian. Okay. David and Florian are two great helpers that I have today. Okay. So how the game is? I need two numbers, David. Okay. So so you practice that, but before that, when when I choose two numbers, then Florian should tell me to put a line, put an age or not, by some rules that head or tail, whatever you assign. Head would be yes, or tail would be no, whatever you want. Just tell me yes or no. Good. So we start the game. So you need to give me two numbers.
2 and 6.
Okay. Should I do anything, Florian?
Yes.
Okay. Oops, not good. Okay, David, what's the other two? What are the other two?
3
Okay.
and 5.
Florian.
Yes.
Nice.
4 and 5. No.
Okay.
6 and 6.
6 and 6? Well, we might have. Yeah, let's have fun. No, okay. No fun. 2 and 3?
5.
2 and 5.
Yes.
Yes.
4 and 2. Yes.
Yes.
3 and 2. Yes.
Interesting.
2 and 4. Got it. Already have?
Yes? Ah, we have already. Okay.
2 and 1. No. 5 and 1. 5 and 1 again.
Okay.
No.
No connection.
2 and 4. We already have.
Yeah.
2 and 1. Yes. 6 and 5.
Okay.
Yes.
Good. Maybe that's enough. Thank you very much. Thank you. So we have a random graph just by. Yeah, go on, please.
I hope you won't give the grades like that.
It depends on the answers. No, I won't give grades randomly, so don't worry. Okay, so let's think about the degree distribution. So the degree of this, the first one is 1. So the second one is 2, 2, 1, 2, 1, 2, 3, 4, 5. Okay, so if I actually draw a distribution, which you may actually do it this way. So you have it to be more transparent. So we can say 1, 2, 3, 4, 5. So how many 1 do I have? This is 1. How many 2 I have? 1, 2, 3.
I think the connectivity of 5 is 3, not 2.
And you have 2 ones.
Ah, sorry. Good point. The rest is okay? Okay.
And then 2, 1.
2 ones, okay. Yeah, note 1 as well. Perfect. So 2 ones, then 2 would be 1, I guess. As 2. 2, perfect. And then 3, do we have any 3? Like 1. Do we have 4? 1. Do we have 5? 1. Yeah, so something like this, right? So if you try this practice many times, what would happen is that you may end up with different forms. But remember, the real shape of your graph would basically depend on the probability. So here the probability of connection is 0.5, is actually very high. We might have cases where the probability is 0.1 or something. And then this distribution would be slightly different. But so this is actually, I wanted to make the concept that this is the way to make a random graph. So two things, the location and whether I should or not connect them. And mathematically, this practice that we did is proportional to a model that was proposed by Erdős and Renyi, some of the great mathematicians. I guess Erdős is probably one of the most well-known mathematicians of all time. And where they were looking at these probabilities. And that actually corresponds to a binomial distribution. So here you have the probability or the chance of making the edge. So you have k, so the degree distribution is k. For each node, you want to make connection with k other nodes from the whole collection of n points. So this is the chance, this is like the head and tail situation that we encountered. And if n is large, so this distribution would actually become a Poisson distribution. And so we know a lot about the mathematics, and we can actually get the degree distribution for some calculations. And just notice that you can have different probabilities here, 1%, 0.1, and 0.5. And based on these probabilities, I can start making random graphs with connections. But that's more or less, if we continue that practice for a long time, we basically more or less get the same picture. And here below, I'm putting the codes that you can actually generate this network yourself with iGraph. Yeah, just see what happens if I increase the probability. So we'll actually get to the network of one of your friends, right? Then you draw the friendship network at the end. So one interesting point is that if n is large enough, if you look at the degree distribution of a random graph, we get more or less like a sort of Gaussian type distribution with an average that is proportional to n times p. So if we have 1,000 nodes and the probability of connection is 0.1, actually this is like more or less on average what you get. So the mean of the degree distribution is 100. So if I give you the plot of a random graph and tell you what's n and what's p, or I don't actually give you p, I only give you, for example, n, and this plot you should be able to somehow guess the probability. And that's interesting because it means that we can look at the graphs and then infer that probability from the shape of the graph. Okay, so it brings us to the next important question, that whether biological networks are random or not. I guess by now we should have some hunches that how would biological networks compared with the random graph. Are they random? Yeah, that's the right answer. So most biological networks are not random. And what you can do is that you can, again, get from the database of interest, you can get a biological network, and then look at the distribution, the degree distribution, and then compare the degree distribution to a random graph. So this is the log-log scale. That's why we get a sort of line here. And well, most biological networks would look like these types of networks often. These are two examples. So for example, this is the case of a transcriptional regulatory network in yeast, and an example of genetic interaction networks, whether two genes would interact. And then what you see is that degree distribution is following a power law. It's not actually a sort of Gaussian or coming from any Poisson distribution. This is another example. So this is substrate network of E. coli. On the x-axis, I have metabolite degree. So whether this metabolite is well connected to other metabolites, and then the rank. So what you see is that, again, we get this sort of power law behavior. So these networks usually have heavy tails. And that is the reason we see this power law. Which brings us to the interesting question that what is so special about these scale-free networks? To give a hint, we often call these networks robust yet fragile. What is robust about them and what is fragile about them?
If you eliminate one of the hubs, the whole network might collapse, but any other metabolite or whatever it is not that interconnected, so not that important.
Yeah, perfect answer. These are robust against random attacks, random failures, because randomly, if you want to do something on this network, attack, changes, mutation, whatever you call, gene deletion, they're actually falling in the majority of the nodes that are not really any hops, so they're not actually much connected. So if you just count them, there's a handful of hops here. However, they're fragile because they're actually fragile against systematic attacks. So if there's something that can actually target the hub, well, the whole system is basically knocked out. And that might be, for example, the first case might be the case of the stability of organisms or genetic circuits towards mutations. Mutations are random most of the time. What happens is that these networks are really robust for single-point mutations or random mutations. However, if there is a situation like cancer, where there's a specific hub that is involved, and we have mutations in that specific hub, maybe this is T53, which is actually one of the genes that is mutated in 50% of all cancers, then the fate is pretty determined. So we have cancer. Another concept which is quite important is the idea of nearest neighbor degree of nodes. So here, what you can think about, so you can actually have an individual node and then think about the degree of the neighbors, right? So here, the degree of this node is 3. So node 2 that is connected to this network has a degree of 5. There are 5 edges. Node 3 has degree of 5, and then node 4 has degree of 2. So what you can do, we can actually make an average of all the edges of the neighbors, right? So you want to see, why do we do this? Why do we care about neighbors? Of course, we should care about neighbors because we are surrounded by neighbors. So we simply want to know what would happen. So what is the connectivity of all the other neighbors that are surrounding me? So that's the idea. We're getting, if you imagine the situation, we're getting into the ideas like compactness of the network. We want to see where our node of interest is within this network. And based on the behavior of this nearest neighbor connectivity, we can define 2 types of networks. So we can have a assortative network. So this is when this average increases with k. What does it mean? If we have a large k, if our node is well connected, and if we increase this k, if you go to the areas of higher connectivity, the neighbors are also more connected. So you're well connected, your neighbors are well connected as well. So you're really living in an area when everyone is well connected. Like equivalent of living in Beverly Hills or something like that. But then a graph can be disassortative if you are well connected, so the node actually has a high connectivity, but the neighbors won't have the same high connectivity, which is normal societies, I assume. This is actually visually very clear. What would be the differences between assortative and disassortative networks? In this case, we have nodes with degree 4, and they're actually surrounded by basically nodes that would actually have high degrees as well. And then here we have a 7, but this 7 is actually surrounded by nodes of low connectivity. So again, this is the concept of hub. We see many, many times that hubs are appearing, whether through different networks or biological networks or simply definitions. But now we have a mathematical way to calculate where is the hub, which is quite cool. So remember, each network is a matrix. So you get the matrix, you can do these calculations, you can find the exact hub of interest. And when your network has 10,000 or 20,000 genes, well, of course, you cannot just do it by looking, so we need to do calculations. Well, this is a simple activity. I don't go through that. So basically, the idea is that how would you make the graph more or less disassortative? Well, this answer might be simple. So if you want to keep the assortative nature or disassortative nature of a graph, you need to preserve hops. So I can add maybe another node. I can connect another node to this hop, or maybe another node here. But if we start connecting, if we start increasing the connectivity of the neighbors by making these sort of connections and edges, then we make the graph to be assortative. And another indication that biological networks, many biological networks are disassortative. So we have this sort of relationship. So the last concept that I'd like to talk about is the measure of graph compactness. It's actually very important. But I'll try to define it as on one of the nodes, and then we actually can do the calculations ourselves. So that is called betweenness centrality. So the idea is, what is the degree of a node compared to all other paths? How well this node is in between the paths in my network. And remember, a path is a set of all edges. So we count the number of shortest paths between two nodes, let's say U and W, and then count the number of those paths that would involve my node of interest. So if I'm interested in node V, we can actually count. So how we can do it? So here, assume that I'm interested in node C, this node. How many paths, how many short paths, what's the number of shortest path that goes from A to B? It's only one. Does this actually contain C? No, 0. So that's actually the idea. So what we need to do is to divide this number by 1, and that actually would be 0. For this path, betweenness centrality would be 0. But it would be more clear if I show you this table. So in the first column, I have all the pairs that I can actually define. So this is the combination of 2 from n nodes. And then I can start calculating. So let's say I'm interested in the path between node A and B, right? Again, we are looking at C, right? So we calculate betweenness centrality for one node of interest. That is the node C in this example. So from A to B, there's one path. Nothing goes through C. 1, 0. That 0 divided by 1, 0. A to E, there's actually one. The shortest path would be this. And would actually go through C. So 1, 1. Then it would be 1. Well, we can do AF, for example. So this is the shortest path, 1, 2, 3. Would also go through C. So then we have 1 and 1, and then again 1. So then you can actually do it for all the pairs and then sum them. So the sum would be 6 here, and that shows that node C has a betweenness centrality of 6. Visually, if you look at this graph, it's clear that C is in between this graph. That is all this calculation is about, basically. But again, we cannot do this sort of visual inferences when we have thousands of points and thousands of edges. So that's the reason we do it. So, well, the very simple question is that which nodes have the highest betweenness centrality here? The blue one, right? Because they're really in between all other paths. And then the red ones are the nodes that have low betweenness centrality. And this is actually a very important concept because many of the essential genes in different organisms would have high betweenness centrality. So here is an example of comparing betweenness, so fraction of basically essential genes. And so we see that more of these genes are within bottlenecks compared to other genes, non-essential genes, basically. So essential genes usually sit in the bottlenecks which have the high betweenness centrality. In cancer, again, so the genes that are often mutated in cancer would have very high betweenness centrality because they are so important for fundamental biological processes. So few advanced topics just to go through in the case some of you may do some projects later with graphs. We can subdivide graphs into communities. You can actually, there's actually a very interesting and sort of intense research field to achieve this. We want to see whether a graph would have different communities. And there are algorithms that would help us to say, yeah, in this graph, for example, we have two communities that are well separated. And this could be hard clustering or soft clustering, so whether basically the overlap would have a higher degree or not. And another important thing that some of you might actually work on later is the idea that we can do calculations on graphs, so it's not only for visualization. We can easily work with chemical reactions, and this is the concept of flux balance analysis. So we have different metabolites, and they are actually connected with different reactions. So we can actually write a set of differential equations for the corresponding metabolic pathway of interest. And then we can solve these equations by different constraints and different conditions. We might say, for example, I am interested to maximize growth, and then solve this set of equations with appropriate methods such as linear programming and the other things. And there are many assumptions that we take in doing this. One of them is that the system is in equilibrium. So there's a steady state, so dmdt is equals 0. But just one point that if you're interested to do calculations on graphs, check flux balance analysis. And so in recent years, what has emerged to be very interesting and exciting is whether we can get all the information from different databases, which you have seen in this lecture, and maybe would also work with them later in Mike's lecture, maybe in the past lecture. So we have different databases that would contain different entities and different information. We can construct and make networks from these databases and try to look at the outcomes, whether we can actually think of diseases as the changes in betweenness centrality, as the changes in degree of these networks. Maybe a mutation that would actually happen in a node that has a higher degree would be more detrimental for your system compared to another node. And this is often called network medicine. So if you're interested, this is a very rich field of work and research as well. Well, I already said whatever I needed to say. Well, networks are interesting, so I hope you come across networks later and do some calculations. Further reading, you can look at these slides. I uploaded the updated slides today on GitHub. So, and then about the exam questions, you will get 5 questions, and these are multiple choices. 2 of these questions are coming from these 3 concepts: random graph, degree distribution, and betweenness centrality. And then we will have 3 descriptive questions that would either come from the nature of the database, what you can find in database XYZ, the types of graphs, and basically different biological networks. So I guess more or less we are done with it. If there's any question, just please ask. Otherwise, I hope you enjoy the lecture. Thank you.